Hey everyone, we are in chapter 9 or 10, chapter 10, slide 30. We are continuing muscle tissue. We've talked about how we talked about the differences between the muscle types, and then we talked about how muscles contract, going over the different proteins that are in myofibrils that make up a sarcomere, going over what happens when calcium is released from the sarcoplasmic reticulum and then binds troponin, moves tropomyosin out of the way so that myosin can bind to actin, so that the thick filaments grab and pull on the thin filaments, and that causes these thin and thick filaments to slide over each other, thus shortening or contracting a sarcomere. What we're going to talk about now is what caused that to happen in the first place. How did the sarcoplasmic reticulum know that it needed to release calcium ion into the sarcoplasm to cause these events? To cause a muscle to contract, you need a neuron, or really electrical stimulation, but to consciously control it, or for your body to control it, it needs neurons to send a signal. Neurons innervate the muscle. They get through that epimysium layer. We'll see a good image of this coming up later in complete anatomy. But neurons, so another tissue type, are talking to muscle to cause them to contract. Pictured in this histology right here, we have a neuron. It's not the whole neuron, it's an extension of a neuron. You'll learn that this is called the axon of a neuron. And then this axon of the neuron branches and comes to several points. And these extensions, these places where the axon ends, it's called an axon terminal. An axon terminal is the ending. That's what terminal means. It's the ending of an axon, which is the last part of a neuron. And this is where a neuron can talk and can communicate with another thing, whether it's a muscle or gland or another neuron. So at this axon terminal, let me erase my little squiggles here so that you can see what's going on here. You can see these dark little dots in this region. Those dark little dots are chemicals, neurotransmitters. That neurons release from axon terminals in order to communicate with something else. So they're storing these chemicals, neurotransmitters, at their axon terminals, neurons are. Neurons release neurotransmitter. Neurotransmitter binds to, in this case, a muscle, and causes that muscle to release calcium, and then calcium causes contraction. The region of muscle, this highly folded section of muscle that is receiving this signal, and by being highly folded, you're increasing surface area for more signal to come in. This area is called the motor end plate. Motor end plate. So an axon terminal releases neurotransmitter onto the highly folded motor end plate of a muscle fiber. The term for this whole region, you'll learn that it's a special type of synapse. A synapse is a region where a neuron communicates with something else. The specific special name here is a neuromuscular junction. It's a special synapse, a place of communication between a neuron and a muscle fiber, neuromuscular junction. Let's take a closer look at what's happening here. As I said, if we look, take a look over here. Here is the cell body of a motor neuron. It's called a motor neuron, a neuron that controls muscle, motor because of movement. So here's the cell body. It's got this really long extension. That's called the axon. That extension is what branches and forms lots and lots of places of communication, and each one of those places is called an axon terminal. And at that axon terminal, you have vesicles full of neurotransmitter. And that's what we were seeing in the histology. Those circular things were vesicles full of neurotransmitter. When an electrical signal spreads down this neuron, and we'll get to that in the nervous system, but when an electrical signal spreads down that axon, we get a signal that then causes those vesicles to be released. Well, the contents of the vesicle be released by exocytosis. So you're going to get this vesicle, for instance, to fuse and then cause the release of all this neurotransmitter. The neurotransmitter can then bind to special receptors. Let's color the receptors blue here. If you remember, we learned about receptors all the way back in chapter 3, I believe, when we learned about cells. Yeah, chapter 3. Receptors are a type of membrane protein. So you've got neurotransmitter, which actually often is a protein or a type of protein. You have a neurotransmitter that's released into the interstitial fluid, binds to the receptor on the motor end plate, and then in this case, it's going to activate a transport protein called a channel. It's kind of like a locked door that you've now opened up. The neurotransmitter is the key. The receptor is the lock. The key fits in the lock. It activates, and now you've opened something. We're opening a channel. This channel lets sodium ion that we're going to assume is floating around in the interstitial fluid, because it is, and you'll learn more about that in physiology. It flows into the cell. We get sodium rushing into the cell. Sodium ion has a positive charge. And when you have a positive charge, or any charge really, but in this case it happens to be positive, when you have a positive charge moving in one direction, that causes an electrical current. Once again, why it's going into the cell, you'll learn about that in physiology. But believe me, for now, sodium is flowing into the cell when this channel opens up. That movement of ion, that is a form of electricity. Any movement of charge, that's electricity. So we've gone from a chemical like a neurotransmitter binding to a receptor. So that was chemical signaling. And now we're getting electrical movement, electrical signaling, sodium ions flowing through this cell. You have neurotransmitter, once again, binding to receptors. Receptors activate channels, which aren't really shown here. So I'm just going to show a channel here and a channel here and a channel here. And it lets sodium in. That sodium, remember, can travel deeper into the cell thanks to those T tubules. That's the function of the T tubule, to help spread the electrical charge deeper into the cell. By having that border, by having that membrane, it can travel along it. It travels down and reaches the sarcoplasmic reticulum. Sarcoplasmic reticulum stores calcium. Calcium, the electrical signal, the sodium flowing down, triggers the sarcoplasmic reticulum to open up its own channels. So there's a different type of channel here, and these channels let the calcium through. Calcium is entering the sarcoplasm. Calcium binds troponin, which isn't shown here. Let's color in troponin. And then tropomyosin is also along the thin filament. It allows tropomyosin to move and shift. By shifting, then the myosin can grab onto actin. That's what's forming here. That's what's going on here. The sarcomere shortens. A lot of steps right there. I'll repeat this again, and if it helps, you should probably rewind. An electrical signal from a neuron reaches the end of the axon terminal. That causes neurotransmitter to be released. Neurotransmitter enters the interstitial fluid by exocytosis. Some of it can bind to these receptors. These receptors are activated. That opens up channels. Channels let sodium ion from the interstitial fluid enter the sarcoplasm. It can travel, the sodium ion can travel deeper into the muscle fiber more evenly thanks to T-tubules. The T-tubules lead this current, this electrical current, down to the sarcoplasmic reticulum. That causes the sarcoplasmic reticulum, it has channels. That electricity causes those channels to open up. Those channels let calcium ion into the sarcoplasm. Calcium can then bind troponin, which makes troponin move tropomyosin. Tropomyosin out of the way now. Now myosin can bind actin. That's the power stroke. That causes contraction. The sliding of the thin filaments over the thick filaments. The sarcomere shortens. I hope that made sense. Please let me know if it didn't. The reverse of this, it's basically to relax the muscle, and muscles have to relax. To relax a muscle, you need the reverse of this. You need to get, the first major thing is you need to get rid of calcium. There's actually other things going on that we're not going to cover, but calcium needs to go back into the sarcoplasmic reticulum. How it gets back there, that's physiology. Sodium needs to get back into the interstitial fluid. We'll talk about that later. And need to remove the neurotransmitter. But we're trying to get everything back to normal, and the sarcomere slides back. So we've seen how one neuron can branch, form many axon terminals. One motor neuron, a neuron that controls muscle, one motor neuron can branch, have many axon terminals that control several muscle fibers. So we see one muscle fiber, oops, let me see a different color. There's one muscle fiber here, there's another here, there's another here, there's another here, there's another here. Those are all controlled by one neuron. The term for this, where you have one neuron controlling many muscle fibers, that's called a motor unit. Where you have one neuron, one motor neuron controlling several muscle fibers, that's called a motor unit. It varies how many muscle fibers one neuron controls. If we want more precise control, then you want to only control 2 to 3 muscle fibers. This is true for something like our eye muscles. To have very, very precise movements, you don't want to have, if you control too many, you're just going to have this really big movement. It'll be really hard to have fine, precise movements. But if you want less precise movements and therefore have lots of strength, lots of power, you can do that with, say, your leg muscles, things like your rectus femoris, which we'll talk about later. But you can control around 2,000 muscle fibers if you want more strength, have it all work together. We've talked about how when things work together or when things are lined up, you get a lot more power. Here, all of your muscles with one neuron are getting controlled. Not all your muscles, all these muscle fibers are being controlled by one neuron. So one neuron gets activated, you activate 1,000, 2,000 fibers all at once. When you see this in your muscles, you usually see that, or it's pretty much always true that one muscle will have motor units of different types. So you'll have like this purple one controlling 1, 2, 3, 4, 5, 6, 7, or you'll have this red one controlling 1, 2, 3, 4, 5, 6, and yeah, they're controlling different ones. So we can activate different ones at different times. Once again, something you'll discuss more in physiology, but the advantage here is that you're doing less work. This is divergence. A path is diverging. You have one neuron to control. That neuron then controls many muscle fibers. A lot less work involved. When we look at the size of muscle fibers, what's interesting is that whether our muscle fibers get bigger or smaller, it's that our muscle fibers get bigger or smaller. It's rarely that we get more muscle fibers or fewer muscle fibers when we gain strength or lose strength. Muscle fibers increase in size due to use. It's not that they undergo mitosis typically. There's a very, how do I put this? It's not fully confirmed, but the hypothesized mechanism is that, let's say you work out. When you work out, you get light damage to your cell. If your muscle fiber is injured, it gets repaired and then it gets bigger. That's the thought behind how we grow muscle. The term for muscle growth, it's called hypertrophy. Hyper means elevated, troph refers to growth. Hypertrophy. So muscle fibers will increase in size. When they increase in size, you're getting water content, so the sarcoplasm is getting bigger, but you're getting more myofibrils, more protein. So that's one reason why people who try to gain muscle mass eat more protein, because you need protein as building blocks to make myofibrils. And we've seen how many different proteins are needed to make myofibrils. You need more mitochondria because you need more energy. And the activity of something called muscle spindles will increase. Muscle spindles, we'll learn, are a receptor for detecting how stretched out your muscles are. So hold that thought, but that's the reason why stronger people, if you ever poke their muscles, feel their bodies, it's harder. The muscles are more tense, more contracted at resting state. It's because of the activity of these receptors, the muscle spindles. The opposite of hypertrophy is atrophy. A means without, troph means growth. So without growth, atrophy. This continued use of muscles causes a decrease in muscle mass, smaller fibers, so basically the opposite of what we just discussed, and decrease in muscle tone. It gets softer. You can even see this with histology. An atrophied muscle has very small muscle fibers. You're not decreasing the number of muscle fibers, you're decreasing the size. You compare that with a normal picture of muscle fibers. You can compare this cross-sectional size of the different muscle fibers. Obviously, the normal one is bigger. They're all pushed up on one another. You don't really have that here for atrophied muscle. Now, there are two types of muscle. Some books will say 3, but we're going to simplify to 2 in terms of what a muscle can do, how it's how muscle is adapted. Sometimes you need muscles to do things for extended periods of time. Like I'm standing here, my body has to be upright. If my muscles weren't contracted to keep me upright, I would just collapse on the floor. My muscles are working to make sure I'm standing up straight. So those muscles need to be working all the time. Those muscles that are working all the time without getting too tired, those are called slow twitch muscle fibers. You might see them called type 1. I'm going to call them slow twitch. Slow twitch muscle fibers are adapted for high endurance, high stamina things that don't require as much force. That's what I mean by low tension. Functionally, they're different. And of course, structure is related to function. Structurally, they are different. We can see that slow twitch muscle looks darker. This is a cross section of muscle fibers. I shouldn't have labeled that as myoglobin. Let me do that again. So there's a single muscle fiber. They appear darker. Look at the difference in color between the slow twitch versus the fast twitch. There's more myoglobin in the slow twitch compared to the fast twitch. Myoglobin is a protein. Sounds a lot like hemoglobin. Globin just means globular, but it actually is related to hemoglobin. Hemoglobin is found in your red blood cells, your erythrocytes, and it's for carrying oxygen. You have it in your muscle cells, your muscle fibers, also for carrying oxygen, this myoglobin. It's a hemoglobin for muscles, myoglobin. It's a way of storing oxygen for when you need it. It's like hoarding oxygen from your bloodstream, which is good for these muscles because you need it to do things for extended periods of time. Myoglobin helps bank oxygen for later for these muscles that do these high endurance, high stamina activities. And therefore it makes it appear darker. The dark meat of a chicken is in the thighs and the legs. What are chickens doing all day? They're walking. They need to stand and walk all day, free range, and move around, and just stay on their feet. So that takes a lot of energy for long periods of time, so it's darker. Another reason why it's darker is that it has a extensive capillary blood supply. You need lots of oxygen, and therefore you need lots of mitochondria. You can see all these gaps in between. That's where blood vessels are going. You see more of that in slow twitch than you do in the other one, the fast twitch. There's a trade-off for everything. If you're going to be high stamina, you're going to have lower tension. These muscle fibers are smaller in diameter, so they generate less force. So, which is fine because these are muscles for posture, for walking. And you can also train yourself to have more of these. Your muscle fibers can switch over. So if you train for marathons, you're going to have a lot of slow twitch fibers. If you train for cycling 100 miles, you're going to have a lot of these slow twitch muscle fibers. I've been pointing to the histology at the bottom. This is a cross section. In longitudinal section, you can see the slow twitch is much darker than the fast twitch. Fast twitch, on the other hand, is the opposite. Fast twitch is adapted for high tension, high force, short burst activities. So it's only for a short period of time where you can use these before they tire out. They are easily fatigued. They're easily fatigued because they don't have as many blood vessels. We saw that in this one. There's not as many blood vessels going in between the muscle fibers. It's less myoglobin, so it leads to a paler color. I can see that here in the longitudinal section as well. And fewer mitochondria comparatively to the slow twitch, which means you're not making as much energy to help sustain for a longer period of time. They are much larger in diameter, though. They're packed with myofibrils. An individual muscle fiber is much larger in fast twitch than in slow twitch. The benefit, of course, is having fast, high power actions. If it's just a few muscle fibers, that's how your eye muscles work, quick darting movements. Your eyes get pretty tired if you're just struggling or reading all day. If you're trying to focus on something, your eyes tire out easily. Power lifters, sprinters will have a high concentration of these fast fibers. And once again, you can train yourself to be more slow twitch or more fast twitch in your muscles by working on more endurance things if you want slow twitch, or working on high power intense things, fast twitch. I was saying that there's actually 3 types of fibers, as you might read. There's an in-between one. It's like we won't talk about it, but we'll just simplify it to slow twitch and fast twitch. So that wraps up our, well, not our entire chapter, but that wraps up this discussion on how muscle fibers are controlled. This animation shows us that an electrical signal passes through a neuron, reaches the axon terminal. You get release of neurotransmitter, activates receptors on the motor end plate, opens up channels, lets sodium in, electricity runs down the T tubules, activates sarcoplasmic reticulate to release calcium. Calcium binds to troponin, moves tropomyosin out of the way. Myosin heads bind to actin. And when they bind to actin, it's kind of like how these rowers grab and pull. There's this power stroke. This power stroke pulls the thin filaments closer to the M line. There's a lot there, so please rewatch those parts where I'm going over the sequence if you need. And please let me know if you have any questions in the comments and discussion. The next video will go over muscle categories, shape categories, fascicles, things like that. So I'll see you in the next video.